Okay, so we are going to start talking about proteins in chapter 5. So remember proteins, these are one of the 4 classes of macromolecules. So remember the 4 classes of macromolecules. There were proteins, carbohydrates, nucleic acids, and lipids. We will spend the most time this quarter talking about proteins. We'll go in the most depth talking about their structure. We'll probably have the most examples of types of proteins. So we're going to start off today with what proteins are, what they're made of, and then talk about the different levels of structure of proteins. So we said when we filled out that chart earlier that proteins are made up of monomers. You can think of a monomer like an individual subunit. So they're made up of monomers called amino acids. The polymer is called a peptide, polypeptide. And then the type of bond that holds them together is called a peptide bond or a peptide linkage. So in this lecture, we are really just going to focus on amino acids, these monomer subunits that proteins are made up of. So there are 20 amino acids that are incorporated in our proteins. There are actually 22 that you are not responsible for those other 2, but the 20 amino acids that you are responsible for are the common 20 amino acids. They can be found in your textbook and in the PowerPoint lecture. There are some proteins that incorporate those other 2 amino acids, but not all proteins require those. So we're only talking about the standard 20 amino acids in this class. So first let's define what an amino acid is, and then we'll start talking about some of the specific amino acids. So an amino acid, it has an amino group and it has an acidic group. So the amino is this amino group at one end. Give this a positive charge if it's NH3. And then it has a carbon in the middle. This is called the alpha carbon. And then it has a carboxylic acid over here. So you'll see it written like this, or you could see it written like this. So either way, and then this would have a positive charge of 1. It has an amino group, and then it has a carboxylic acid group. So that's where the name amino acid comes from. It has an amino part and an acidic part. So something else that's important about an amino acid, I'm just going to draw it again down here. Is this carbon right here. This carbon always has a hydrogen on it, and then it has something else bonded to this carbon. So this is kind of the generic skeleton structure of all 20 amino acids. What makes every amino acid different is this, what we call an R group. So there are 20 R groups for the 20 amino acids. So all 20 amino acids look like this. They just have a different R group there. This carbon right here that is not part of the amino group or the acid group, this carbon that has a hydrogen attached to it and also the R group is called the alpha carbon. So this is the Greek letter alpha. So it's important that you understand this alpha carbon location, because when we're talking about structures of proteins and specific amino acids, we will orient to this alpha carbon. So this carbon that's part of the carboxyl group is not the alpha carbon. It's this carbon in the middle, nitrogen, carbon, carbon. This carbon here that is attached to the R group. So there are 20 amino acids with 20 different R groups, and these R groups are all listed in your textbook. It shows all of their different shapes. They're broken into groups: aliphatic, nonpolar, aromatic, polar, positively charged, and negatively charged. So it'll be important to understand why these amino acids are grouped together. They were going to act similarly. Negatively charged amino acids will act like other negatively charged amino acids. It'll be really important to know if an amino acid is polar or non-polar. So these properties are important to understand for an amino acid. So here you see in green, this is the location, this is the R group, and you'll see these 20 amino acids are all different. They all have a different R group here. Glycine is an exception to all the other 19 amino acids because glycine has an R group that is another hydrogen. So that means glycine is the only amino acid that is achiral, meaning it's not chiral. The other 19 amino acids are chiral. And that's important. This is going to have a lot to do with the structures of the proteins and the types of conformation. Conformation is just a word that means shape. The shape of these folded proteins. So the amino acids, the sequence of amino acids is the first step to getting a functional protein. So these are the 20 amino acids. You will need to know these. You will need to know the properties of them. You'll need to be able to identify them or draw them. And they all have this basic skeleton structure with a different R group. So let's just say all amino acids are chiral. They have this chiral stereocenter here at this alpha carbon, except for glycine. Because glycine has an R group that's a hydrogen. So this carbon can't be chiral because it has two substituents that are the same. For the amino acids also, you'll have just using glycine as an example here, there's glycine, there's the word, then there's the 3-letter code, which for glycine is GLY, and then there's the 1-letter code, which for glycine is G. So you'll see amino acids referred to either as their full name, their 3-letter code, or their 1-letter code. You can find all of those in your textbook, and you should be able to use any of those. Okay. So you'll notice that this amino acid has an overall charge of 1 because this over here is neutral, this carboxylic acid, and then this amino group has a charge of +1. So I would say that the overall charge of this amino acid is +1. So most amino acids are written in what's called their Witter Ion form. A Witter ion is when the amino acid is charge neutral, meaning it has an overall charge of 0. So this happens when the pH is at what's called the pi, or the isoelectric point of the amino acid. And all 20 amino acids have a different isoelectric point. And we'll talk about isoelectric points in a little bit, but this is the pi. Okay, so if I was to draw the Witter Ion form of glycine, it would look like this. Okay, so this is glycine and this is the Witter Ion form. So here I have the amino group. Here again, this is my alpha carbon. This is my R group. Because this is glycine, I know it's just a hydrogen. And here is my carboxyl group that has lost a hydrogen, which now has a charge of -1. So this is the Witter Ion form where we are protonated here and deprotonated here. So another way that we'll describe this in biochemistry is if we have a positive charge, that's called protonated. And when we've lost a hydrogen, this is called deprotonated. So this is the Witter Ion form of glycine. Back to when we were talking about all amino acids being chiral except for glycine. So if you have structures that are chiral, that means they can exist in different isomers, different enantiomers. So all amino acids except glycine are L isomers. So this would be L whatever amino acid. All amino acids that are incorporated to proteins and living things are found with the L enantiomer. L is what we use in biochemistry. In organic chemistry, L equals S. R is D and L is S. So these are all L amino acids, but if you're used to assigning something from organic chemistry, this would be S. We'll do everything with L and D. D is the same as R. So there's no D amino acids. They're all L amino acids in proteins in living things. Okay, so let's talk a little bit more about this Witter Ion. So all amino acids have a titration curve because they all have acidic protons. We just saw there was one right here. So let's just look at the titration curve of glycine since we just drew this. So this also can be found in your textbook. So if you're looking at the titration curve of glycine, it's going to look something like this. Where we're going to have a pK1 here and a pK2 here. So these are for the two titratable hydrogens. So there's two titratable hydrogens. If we come back to over here, there's two titratable hydrogens, meaning two hydrogens that can leave. The first titratable hydrogen is going to be one of these three hydrogens on the amino group. And the other titratable hydrogen is going to be this hydrogen on the carboxyl group. Down here at pK1, this first hydrogen that's going to leave is going to be this carboxyl group. So all carboxyl pKs are around 2. So here for glycine, it's 2.34. The pi, which we'll talk about in a second, is 5.97, and then the pK2 is about 9.6. So this is the PK1. This is of the carboxyl group. And this PK2, this is for the amino group. And here in the middle, this is the isoelectric point. This is the PI. This is where the Witter Ion is. This is where we have that, both of those. Where we've lost the hydrogen from the carboxyl group, but we still have the hydrogen for the amino group. So I'm just going to go ahead and show you what this looks like because it is a little bit bigger here. You might be able to see it a little bit better. So this is the same titration curve that I just drew. It's just showing what is happening in each of these situations as the pH changes. So here we have a glycine. We know it's glycine because the R group is just a hydrogen. We'll use glycine as an example a lot just because we don't have to draw whatever structure of the R group that the other ones have. So here's my amino group, alpha carbon, carboxyl group. At a low pH, lower than the PK1, this carboxyl group is protonated, meaning it still has its hydrogen. So that means the overall charge of this amino acid is +1. Here, at a pH that is between the PK1 and the PK2, once we are above, at a pH above this PK1, we will lose this hydrogen. That hydrogen will be donated. The acidic proton will come off at a pH higher than the PK1. So this is still glycine because of that hydrogen. So I still have the hydrogen here on the amino group and 3 hydrogens, which gives it a +1 charge. I've lost the hydrogen from the carboxyl group, so now this is a Witter Ion. This is a neutral amino acid because we're existing at a pH between the PK1 and the PK2. So most physiological pHs fall in this range between the PK1 and the PK2. So most of the amino acids that we're going to see at a physiological pH of around 7 are going to be Witter Ion forms. Here, once we are above the PK2, so at a pH above the PK2, now we're deprotonated. We've already lost the hydrogen from the carboxyl group. Now we've lost one of these hydrogens from the amino group. Now this is no longer positively charged. It has no charge, and we're still negative over there on the carboxyl group, which means this has a charge of -1. So you're going to hear some terminology about amino acids being protonated or deprotonated. This has to do with the pH and if you are above or below the PK1 or the PK2 of each amino acid. So there's a table in your textbook. It's actually this table. It's table 5.1. It shows all the amino acids, their 3-letter codes and their 1-letter codes. It shows the pKa of the carboxyl group, the pKa of the amino group, the pKa of the side chain, if there is one, and then it shows some other things, size and occurrence in proteins. You don't need to memorize this because you'll always have access to it, but if I asked you to draw the structure of glutamine at a certain pH, you would be able to tell me if these side chains are protonated or deprotonated based on if you are above or below the PK1 or the PK2. So this is the PK1, the carboxyl group, PK1, and this is the PK2 of the amino group. Some have a PK, another PK if there is another titratable hydrogen on a side chain. We won't talk about any of those today. But that's the deal with amino acids. We will talk about putting amino acids together with peptide bonds to form polypeptides next. But you should be familiar with the 20 amino acids, their 1-letter code and their 3-letter code, and also be able to interpret a titration curve for the amino acids and tell me what the structures of the amino acids look like at different pHs.